numbers and cell viability using a variety of different approaches. There are direct cell count methods and viable cell count methods. Direct cell count methods include little counting chambers that you can look at under the microscope, and we'll do a bunch of this if you take my wine microbiology class, for example. Electronic counters that actually can count little microbes that you either label, for example, or with a dye of some kind, and then there's a little laser that can count them as the microbes are shunted through a little conduit of some kind, and then membrane filters, which I'll show you. So a way to pull microbes down and capture them on a filter, and then take the filter and physically look at that under the microscope, and you can do direct counts that way. The viable cell counts include various plating methods and membrane filtration methods, which I'll mention in the next slide. So here's a membrane filtration method. And so in this particular case, you've got a membrane filter apparatus and a water sample, for example, that's run through these 0.45 micron filters. And then, and you can have smaller filters than this that could catch smaller microbes, because some microbes, could pass right through that. Anyway, you grab this filter with sterile tweezers and apply that to a petri plate containing some kind of nutrient media. It could be, selective or differential for the growth of certain kinds of microbes, and then let them grow over a period of time, and you can do a direct quantification. So for example, if I put 100 milliliters of fluid through that sample and got 1, 2, 3, 4, 5, 6, 7, 8, 9 colonies, I can say that there are 9 cells per 100 milliliters and get an estimate of the concentration. Here's more of a realistic look, and this is an approach for detecting fecal bacteria in samples. And so in this particular case, you see the gridded filter that was placed on top of this petri plate, and the petri plate contains selective and differential ingredients that can allow you to differentiate different kinds of microbes in a water sample. And so these might be, for example, E. coli, these blue colonies. And so if you're really interested in looking at E. coli counts in water samples, you can use an approach like this. Obviously, the environment has a tremendous amount of impact on how microbes grow. Most microbes just prefer fairly moderate environmental conditions. So basically, whatever you like, a lot of microbes prefer that as well. The moderate temperatures and moderate sun exposure and all that stuff. So again, whatever you think is comfortable, a lot of microbes prefer as well. But there are these extremophiles that grow under really harsh conditions that would kill most other organisms. So extreme environments can be a deep sea hydrothermal vent or the hot springs in Yellowstone or whatever, things like that, or the Great Salt Lake in Utah or the Dead Sea, things that are super high in salinity. And so a lot of the characteristics of microbes are broken down in this table here. So you have descriptive term, definition, and the representative kinds of microbes that might fall into those categories. And so over here you see microbes that are osmotolerant, meaning that they're tolerant of inhospitable osmolarity. And so they're able to grow over a wide range of water activities. And what that means is that they can grow over a wide range of, for example, salinities. And shouldn't surprise you that a microbe like Staph aureus, which lives on human skin, which can be very salty, can handle a wide range of salinity. Halophiles are microbes that specifically love high salt. And so osmotolerant organisms, the suffix tolerant means the organism can handle it, but it's not ideal. So they're tolerant of osmotically unfavorable conditions. But a halophile, on the other hand, is somebody who loves salt. So phile means loving, and halo refers to salt. So again, and these are organisms that can live in the Great Salt Lake in Utah or the Dead Sea and so forth. Acidophiles, an acid-loving microbe, and so their pH range is in the acidic range. A neutrophile, these are organisms that love temperatures that sort of hover around neutral. An alkalophile are organisms that prefer alkaline. And then if you go down to temperature, psychrophiles love the cold. And psychrotrophs. Psychrotrophs is a weird term because it's, again, it's a modification. The suffix is a modification. So psychrotolerant or psychrotroph, meaning microbes that can handle the cold but prefer to grow at warmer temperatures. And you can see that here. So a microbe that can handle the cold but prefers to grow at the warmer temperatures. And these guys are particularly problematic because these are microbes that can grow in the refrigerator, for example. They don't like it, but they can grow in the refrigerator. And then if you ingest something that they've grown on that came from the refrigerator, they can grow even faster in your gut tract. Mesophiles are middle temperature loving. Thermophiles are hot temperature loving. Hyperthermophiles are extremely hot temperature loving. And then there's different microbes that are broken down by their preferable oxygen concentration, like an obligate aerobe that prefers atmospheric oxygen concentration. Facultative anaerobes are microbes that don't require oxygen, but do better in its presence. So if you give them two opportunities, one aerobic and one anaerobic, they'll clearly migrate to the aerobic environment. But they won't die if they're stuck in the anaerobic environment. They have the flexibility of metabolism. Aerotolerant anaerobes are microbes that can grow equally well. It doesn't matter whether there's oxygen or not. But the point is that they are technically anaerobes because they don't use the molecular oxygen for anything. And then the obligate anaerobes are microbes that have to be in the presence of oxygen, have to be in the absence of oxygen and can't tolerate the presence of oxygen. They actually die in the presence of it. Microaerophiles, small air loving. So these are microbes that grow in, something less than the 20% atmospheric. Amazingly, many of the microbes in your mouth are microaerophiles that live just below your gum line. And then lastly are barophiles. So baro referring to pressure loving. So these are, deep sea microbes, for example. Water activity is a reference to the amount of water that's available for a microbe's metabolism, and it can be reduced by interaction with solutes. So the more sugar, for example, or the more salt you push into solution makes this situation more difficult. It reduces the water activity. It reduces the availability of water for a microbe. And so that's another way to reduce water activity is basically to just dry something out. So a cracker, for example, has very low water activity, and therefore you can leave a cracker on the countertop for a week or two and it's not going to get all moldy and fuzzy. Meanwhile, if you left something with a higher water activity, like a piece of ham or something, something wet, like a piece of fruit, that's wet and sugary, that'll obviously turn fuzzy pretty quickly. So this slide is giving you again an overview of different water activities and the environments and the types of microbes that can tolerate these things. And so pure water has a water activity of 1, again 100%. And then bread has a slightly lower water activity. Ham has a still lower activity. And as you start to get down the list here, things get drier and drier, like cereal and chocolate and honey and dried milk. And these are the more inhospitable environments that have the longer shelf life. Regular fish as opposed to salted fish. Regular fish will stink and turn foul pretty quickly, but if you salt fish, it can last quite a bit longer. Again, because the environment's inhospitable for microbes that can't handle that extra loss of water activity. So this is just looking at sodium chloride effects on growth, and here on this y-axis is growth rate, some arbitrary growth rate, and here's concentration of sodium chloride, and you're basically looking at a couple different kinds of microbes relative to their salt tolerance or lack thereof. So again, here's a non-halophile microbe that just drops right off the face of the earth in terms of growth when salinity is raised slightly. Here's a moderate halotolerant organism that can kind of handle higher salinities, but as you can see, it's only tolerant of them, and therefore its growth rate rapidly declines as concentration of salt increases. And here's a moderate halophile, and then lastly an extreme halophile that only starts growing when the concentration of salt hits a certain high level. And we also break microbes down into pH, as I talked about already: acidophiles, acid lovers, neutral lovers, and basic lovers. So how do these microbes deal with these acidic or alkaline environments? Well, most of them maintain a near internal pH of near neutrality, which is really interesting. Even if you're living in an acid environment or an alkaline environment, they tend to try to keep everything near neutral. And so that means that they have to keep their plasma membrane relatively impermeable to protons. They can't just let a whole bunch of protons in and acidify the cytoplasm, for example. And a lot of these microbes have proteins that they use to protect their DNA, for example. And so these acid shock proteins that glom onto the proteins or glom onto the DNA to protect them from destruction. Another cool thing that microbes do is they can change the pH of their habitat by producing acidic or basic waste products. And so again, that's a common concept in microbial biochemistry, is the ability to identify microbes based on the types of acidic or basic waste products that they produce. And we'll do some of that in the lab. As far as temperature goes, there's these cardinal temperatures: minimum, maximum, and optimum for a microbe. So again, on the y-axis is growth rate, on the x-axis is temperature. And so here's an organism's minimum, and so its growth rate is going to be pretty negligible. But as the temperature increases, the growth rate ramps up significantly until it hits an optimum. But what's interesting about microbes is they fall off fairly quickly after their maximum is reached. They don't just gradually sort of decay. They actually fall right off. And so they have very specific optima, and then a couple degrees more than that, and they can actually start to die. So here's the growth rate graph for a variety of different kinds of microbes. Again, here are the psychrophiles, the cold lovers that tend to have an optimum around 9 or so, or 10 degrees Celsius. The psychrotrophs, remember, these are the organisms that can handle cold temperatures and grow, but they prefer to grow, it's something closer to room temperature or body temperature. Mesophiles are most of the human pathogens. These are microbes that have an optimal temperature right around human body temperature. And then, of course, the thermophiles that can grow at really warm temperatures, really hot temperatures, actually. I mean, you wouldn't really want to be sticking your hand in these temperatures, especially for very long, especially down here for very long. It'd be pretty uncomfortable. And then hyperthermophiles that shift their temperature tolerance even further to the right. So some of the adaptations of thermophiles, they prefer more hydrogen bonding in their protein structure to keep their proteins together and keep them from unfolding and denaturing. There's lots more prolines. Prolines are cyclic amino acids that have a good solid temperature tolerance. And chaperones are proteins that microbes will use to stabilize other proteins, keep them from unfolding. And then histone-like proteins that stabilize DNA. And then their membranes can be stabilized by a variety of other means, like more saturated or branched high molecular weight lipids, and then the ether linkages that I mentioned before in the archaea, as opposed to the ester linkages in the bacteria. Here's a way we experimentally demonstrate oxygen tolerances for different organisms with this certain kind of media. This medium contains a chemical that reacts with molecular oxygen, and so there's going to be a fair amount of molecular oxygen at the top, and then gradually less and less as you move deeper into the tube. So if you inoculate an obligate aerobe into this reducing medium, these little green spheres represent the microbial growth. The bacteria will grow really heavily at the top. These microbes, the facultative anaerobes, however, will grow more densely at the top where there's oxygen, but they can grow all throughout and get gradually less concentrated towards the bottom. But again, these guys have the ability to shift, the ability to shift metabolism. The aerotolerant anaerobes simply ignore oxygen, and they grow all throughout a medium, whereas the strict anaerobes only want to grow down at the bottom where there's no oxygen. And then, of course, the microaerophiles, the small air-loving microbes, will grow just below the surface where the oxygen content is a little less than ambient. Some of the basis for oxygen sensitivities include detoxifying oxygen radicals. So a microbe that's able to do this can tolerate oxygen. So you and I, as aerobes, have these types of chemicals within our bodies, these enzymes that allow us to detoxify radicals from oxygen metabolism. Oxygen's a pretty messy, toxin-ridden fuel, and so we have these things so superoxide radical is a toxin that our aerobic respiration apparatus makes. And so we produce an enzyme called superoxide dismutase. Microbes do too. Well, not all, but air tolerant microbes make superoxide dismutase, which breaks down this radical. We also produce catalase, and so do microbes. And catalase breaks down hydrogen peroxide, for example. And so getting rid of hydrogen peroxide is important. So if you've ever taken hydrogen peroxide, for example, and swish with it, put a cap full of it in your mouth and swish it around, you'll see bubbles form. And those bubbles are actually formed by catalase production in the microbes on your teeth. They're taking the hydrogen peroxide, using their catalase enzyme to split the hydrogen peroxide into water and oxygen. So the bubbles are actually the oxygen that's being released by the catalase producing microbes. As far as other ways to inhibit or kill microbes, radiation is a great one, and ionizing radiation is an example of that, where X-rays and gamma rays can be used to cause mutations and death in microbes. But these things have such high energy that they can actually disrupt chemical bonds as well, and not only cause mutations in DNA, but just blast through DNA and totally shred it. And not only DNA, but proteins and membranes and everything. So ionizing radiation is a great way to sterilize things because it's highly penetrating as well. My sister actually works at a company that emphasizes ionizing radiation, and they sterilize medical devices, for example. So you could package a catheter and put it in a box and then put it in an ionizing radiation sterilizer and sterilize it and ship it off to the consumer. So biofilms are another way microbes live and grow, and they're ubiquitous in nature, and they're basically just slime-enclosed colonies that are attached to surfaces. So the microbes will actually produce polysaccharides, proteins, and DNA that form a gum, essentially, that keeps the microbes stuck to a surface. So think about the enamel of your teeth, these complex slime-enclosed colonies of microbes on the enamel of your teeth that you actually have to brush with a toothbrush or scrape, go to the dentist and actually have them scrape them off with a metal hook. They often form on medical devices and implants that can lead to severe illness, even water pipes, lots of different places. And that's why I was saying before, to get rid of these biofilms, you often have to use something like ionizing radiation. So what's really fascinating about them, though, is that they're complex interactions among a bunch of different kinds of microbes that are attached to the surfaces. And so these complex interactions that involve cell-cell communication and so forth so that the microbes can coordinate their biofilm excretion. And then they share their gum together, essentially, and use it to seal themselves into these self-contained environments. The amazing part about them too is they actually protect the cells as a group from harmful agents like UV light and antibiotics. And it's one of the reasons why sometimes antibiotics don't cure bacterial infections in people, because if the microbe has formed a biofilm on a heart valve or somewhere in the tissues, it's the slime layer protects them from the antibiotics. Even things like UV light, which can be pretty damaging to cells, that's non-ionizing radiation, can actually cause damage to DNA, just like it does to you and I. We go out in the sun. If we get too much sun, we can get melanoma. Well, bacteria also get damaged DNA, and it can kill them. So, but in a biofilm, they're less likely to be killed if they're hiding hiding out like that. And so the picture looks something like this, where you've got some kind of a substrate that's preconditioned with ambient molecules, and these could be nutrients or who knows, all kinds of things, but charged particles or non-charged particles, but mainly food that the microbe would want to migrate towards, and you can see them either swimming actively to it or depositing on the surfaces. They adsorb to the surface, and there you see them forming a sort of a microcolony. They can pull themselves off and swim away, but typically what they'll do is they'll hang out if the conditions are right, if there's nutrients to metabolize, and they'll start growing. And here you see them growing, but they're also communicating with each other, the cell-to-cell signaling, and that's triggering exopolymer production. And then eventually more cells grow and more nutrients diffuse in and more nutrients deposit, and the microbes start to form these huge piles. And again, these can be on the enamel of your teeth, they're on a scrubby looking rock in a stream or wherever.